Hello.
Try again. Can you hear me now?
Yeah, hello. Yes, yes. Hello.
Okay.
Happy New Year.
There we go. Welcome everyone to the Centers of Excellence in Hematology, the Utah Center for Iron and Heme Disorders seminar series. We want to welcome those who are joining us. Either you've been here before or you're joining us new. We're very excited today to have Yoon-A Kang speak to us. Before I introduce her, I want to just take a minute and share my screen and remind you of our website. So let me get to that. Can you see that screen? That's our website. So this is our website for Utah Center for Iron and Heme Disorders. And if you simply scroll down this page, if I can do it, you'll see here's our team. We have a postdoc student exchange program that's coming up in March. And if you have questions about that, feel free to reach out to me. We actually have a YouTube channel where we post these virtual talks so that if you didn't get a chance to hear it, or if you want to go back and re-listen to it, you can find them by following this link right here. We feature faculty members and publications that happen. Importantly, and what I really want to draw your attention to are funding opportunities. So if you go on the funding link here, right now, Utah doesn't have a funding call, but the Oversight Center has a funding call. So if you click through here, you'll get to the coordinating centers funding opportunities. These are pilot and feasibility opportunities. We have a call for type B grants that's due February 16th. And if you go to the cceh.io page to funding, you can read a lot of details about what these are. There's a type A grant and a type B grant, and they're up to $75,000 for a year of science. And the type A's give you monies that you can use at cores that are available around the country. And you can scroll down this, learn how you can apply. You're also welcome to reach out to me. You can look and see previous members who've received funding. And so if you're looking for opportunities for funding, I highly recommend you take a peek here at this site. And then I'm going to stop sharing my screen like that. And then I just want to introduce Dr. Kang to you. Dr. Kang comes to us. She is an assistant professor in medicine at, at Wash U in St. Louis. And she got her bachelor's and master's degree in Korea. And she got her Ph.D. at the University of Wisconsin-Madison in Emory Bresnick's laboratory. And then she went on to do 2 separate postdoctoral fellowships, where she was very productive in working on stem cell biology and myeloid inheritance. And today, she's going to talk to us, and the title of her talk is Myeloid Lineage Specification Mechanisms at the Top of the Hematopoietic Hierarchy. And just one other funny thing, today is the first time I've met Yoon-A, and it turns out we are published together on a paper. So that was kind of fun to discover. So Yoon-A, thank you so much for talking today. And we will let you populate questions in the chat for those of you who are here. I will monitor them, and we'll probably hold them to the last unless I see something that's an extremely important question during the presentation for clarification. So if you'll just hold your questions to the end, that would be wonderful. And thanks to all of you for joining, and thank you, Yoon-A, for presenting today.
Okay, thank you. Okay, I will start sharing my slide. So hopefully everybody can see my slide, I guess.
Looks good.
Oops, sorry. Yes. Yeah, okay, so good morning, everyone. Again, thank you very much for inviting me to share my story, this CIHD seminar series. I'm very happy to be here today. And I'm a relatively new PI at the WashU Hematology Division, and I'm studying more like, to be honest, like cell-fate decision using the hematopoietic system. So in the hematopoiesis, I mean, already a lot of in this audience know, but I will start with a brief introduction so we are all in the same page. So basically, hematopoiesis is a process of making whole mature blood cell from the lifelong self-renewing hematopoietic stem cell. And then this HSC will further differentiate into the multipotent progenitor population, and then they will keep going more lineage-restricted progenitors such as megakaryocyte erythroid progenitor MEP and granulocyte macrophage progenitor GMP and common lymphoid progenitor CLP, and then they will keep differentiate into the fully functionally mature blood cell. So I mean, people already know that this canonical hematopoietic tree doesn't really reflect what really happening in our body anymore, because there's a lot of bypass mechanisms. So for example, erythroid cell or the megakaryocyte cells are actually coming directly from the HSC, and this like lineage restricted MEP or GMP, this concept is also become a little less clear anymore. So clearly the lineage specification, now the current view is starting from this HSC and MPP population. So let me give you a little more introduction about this MPP, because clearly people have a less so far didn't really pay attention too much at the MPP until recently. So MPP, now around like 10 years ago, the Camargo group at Harvard using the Sleeping Beauty transposon system, and then showing that actually MPP is a major contributor for the blood production in the native hematopoiesis. I mean, previously we always used the transplantation system, which involved a lot of inflammation. So in that context, HSC was the one who can produce all the lineage mature cell for the lifelong. That's true, but like we didn't know that was in the unperturbed steady state hematopoiesis. And then this data clearly showing that, so each line is actually each unique barcode, and then you can see that a lot of mature cell barcodes share with MPP and downstream myeloid progenitor, but barely with a long-term HSC, clearly showing that in the native hematopoiesis, this MPP is a major contributor. And another interesting is actually we now know that even this MPP is not the homogeneous. It's a very heterogeneous population, and there is a functionally distinct lineage bias MPP subset, such as megakaryocyte bias MPP2 and granulocyte macrophage bias MPP3, and then lymphoid bias MPP4. Important thing is this lineage bias MPP population, they are all independently produced by HSC, which means now if we think about how this blood system makes a certain lineage of cell, we have to think about first how the HSC decide to make this lineage bias MPP subset, and this MPP's lineage bias MPP subset within the population, how they decide to make one cell type versus the other. Because as the name implies, although I said that, for example, MPP3, they are making granulocyte macrophage, they like to make those, but they can also still make the erythroid. So to really see that how the cell decide to make the granulocyte, for example, versus erythroid, we need to know that in the MPP3 level, how they decide to go to GM versus the erythroid lineage. So that's why I'm focusing both HSC and MPP population to really map out how they cells decide to make one cell type versus the other. So this is basically immunophenotyping scheme, how I distinguish this lineage bias subset MPP2 and MPP3, but I'm not gonna go into the detail here because probably a lot of people already familiar with this staining scheme. So during my postdoc, I was kind of focusing on the myeloid lineage specification. So in the steady state, the HSC usually make a relatively small number of MPP2 and MPP3, and then a lot of lymphoid bias MPP4. And these lineage bias MPPs work together to make all the balanced lineage output. However, upon stress condition, HSC transiently overproduce this myeloid bias MPP2 and MPP3, and even they molecularly reprogram this lymphoid bias MPP4, which will overproduce the GMP, which lead to the overproduction of the granulocyte macrophage. However, in this stress condition, the stress is gone, then of course they come back to the steady state because they maintain their homeostasis. And in the leukemic condition, unlike the stress condition, now HSC constitutively overproduce this myeloid bias MPP2 and MPP3, and then MPP4 is also constitutively making the more myeloid cell, which will overproduce myeloid lineage, which lead to the myeloid leukemia. So the question is then, okay, then how does HSC decide to make this myeloid bias MPP2 and 3 versus lymphoid bias MPP4? So I started looking at the signaling pathway. It's well-known, functionally well-known in the HSC, such as Notch, Wnt, or Hedgehog, and TZ-beta. And I'm just showing the little kind of representative gene expression profile. Then you can see that interestingly, this Notch and Wnt signaling have a kind of opposite expression pattern, which means they are relatively high in the MPP4 population, and then Wnt signaling is more relatively high in the MPP3, kind of make me think, oh, maybe when HSC has a high Wnt signaling, they make more MPP3, and HSC has more Notch signaling, they making more lymphoid bias MPP4. So to test this in the cellular level, I used the HES1 GFP reporter mice. And then clearly you can see that upon DLL1 Notch ligand stimulation, actually HSC has most high signaling activity upon this stimulation, followed by MPP4, and the MPP3 barely has Notch activity, as shown by here, the quantification. Then what about the Wnt activity? So I use the nuclear beta-catenin immunofluorescence so as a measure of the Wnt signaling. And you can see that clearly MPP3 has most robust nuclear beta-catenin level, indicating MPP3 has most high Wnt activity, and then followed by HSC, then MPP4. So this cellular data really showing that, oh, really indeed, the MPP3 has a high Wnt, and the MPP4 has a high Notch signaling. Again, to make us think that, oh, when HSC has Notch signaling, high Notch signaling, they will probably making more MPP4. And then when they have more Wnt signaling, they will make MPP3. And it kind of makes sense in the steady state, because I just show you that in the steady state, HSC has most high Notch activity and then low Wnt. So that's why they making more lymphoid bias MPP4 a lot and relatively small number of myeloid bias MPP3. Then I want to check that whether really indeed changing this Notch and Wnt signaling, we can modulate the production of this lineage bias MPP population. So first, because as I say, I'm more interested in the myeloid lineage, I want to check that whether if I lower the Notch, can I increase the myeloid bias MPP production? So I use Notch1 conditional knockout mice, and when I reduce the Notch signaling, then MPP2 and MPP3 is expanded. And when I increase the Wnt signaling using the beta-catenin gain-of-function mice, again, I can increase MPP2 and MPP3, clearly showing that by modulating this Notch and Wnt signaling, I can modulate the production of this different lineage bias MPP population. Wait, but then I think I'm using my cursor, but is it showing on the presentation, or should I make the pointer?
It would be better if you made the pointer. You could,
okay.
Yeah, that's better.
Okay. Yeah, sorry. I thought I was using my cursor, so I thought it's showing, but now maybe it wasn't. Sorry. I will now keep using this pointer.
Great.
So basically, yeah, this data showing that, yeah, indeed, in the molecular level and then cellular level, and then indeed, in the in vivo mouse model, when we increase the Wnt signaling, we can increase this MPP3 production. So again, as I say, in the steady state, because HSC has a high Notch activity, they're making more lymphoid bias MPP4. And then because they usually have relative Wnt signaling, the myeloid bias MPP3 is low. Then I wonder whether in the leukemic condition, do they use the same signaling pathway to modulate this production of different lineage bias MPP, or they're using something else? So to answer this question, I use the chronic myeloid leukemia CML mouse model. So CML is basically caused by constitutively active tyrosine kinase BCR-ABL transformed the HSC. And because it's a very slowly progressing myeloproliferative neoplasm, it's for the researcher, it's good to use as a model system because, for example, AML, the mouse model, they die so fast. But this CML mouse model, they slowly progressing. And importantly, also, they have the whole spectrum of lineage specification for the myeloid lineage from HSC to MPP3 and GMP and mature granulocyte macrophage. So it's a really good model system to study myeloid lineage specification. So I use this CML mouse model, well-established CML mouse model as CLTTA, TRA B-cell able. So from now on, I will call this as a BA mouse. So using this BA mouse, I asked that whether in the leukemic stem cell, they have different notch signaling. So again, I use a HES1 GFP reporter and then cross with the BA to make the leukemic stem cell. And then when I stimulate with the notch ligand, clearly you can see that compared to control HSC, leukemia HSC has less notch activity shown by this quantification. So showing that lower notch activity in the leukemic stem cell compared to control HSC. What about Wnt signaling? Again, I did the nuclear beta-catenin immunostaining, and then compared to control, the BA HSC has way more nuclear beta-catenin level, indicating they have high Wnt activity. So this data clearly showing that in the steady state, unlike the steady state in the leukemic stem cell, they have a relatively notch signaling and high Wnt signaling. That's why they overproduce the MPP and then relatively less MPP4. So clearly showing that leukemic stem cell basically hijacking the normal inducible myeloid regeneration pathway, but then just keep constitutively activating those regeneration pathway to overproduce this myeloid bias MPP3, which will lead to the myeloid disease condition. Then I asked whether by modulating this notch and Wnt signaling, so basically by dampening this constitutively activated myeloid regeneration pathway, can I attenuate the disease condition? So first, to increase the notch signaling, I crossed the notch1 intracellular domain expressing mice with the BA mice. So in this BA NICD mice will have a higher notch activity in the leukemic stem cell. So you can see compared to BA mice, the BA NICD mice, which has higher notch signaling, these mice clearly have the survival benefit, showing that by modulating this notch activity, which means dampening this constitutively activated myeloid regeneration pathway, can attenuate the disease progression. Then what about the Wnt signaling? So to reduce the Wnt signaling, I crossed the BA mouse with the beta-catenin conditional loss of function mice. And I know this is a very kind of complicated scheme, but basically I reduce the beta-catenin level before I induce the disease model and then follow how they are doing. And similar to the notch increased mice compared to BA, these Wnt lower BA mice have a survival benefit. And then I'm not showing all the data, but basically they have a reduced MPP3 expansion and then reduce the GMP and then mature bone marrow cell. And even HSC level, their lineage output without the massive cell load, showing that they have a reduced myeloid colony formation. So again, this data really showing that by modulating this notch and Wnt signaling, we can modulate this lineage bias MPP3 expansion, and then it lead to the attenuated this myeloid malignancy. So if I summarize in this part 1, so in the steady state, HSC has high notch and low Wnt signaling. So they actually produce more lymphoid bias MPP4 and relatively small portion of myeloid bias MPP2 and 3, which will make it all the balanced lineage output. However, in the stress condition, HSC, they have a low notch and relatively high Wnt signaling. So transiently expand this myeloid bias MPP2 and 3, and then molecularly reprogram this lymphoid bias MPP4 toward the myeloid lineage to produce overproduction of a myeloid cell. But in the stress condition, once this stress is gone, they maintain their homeostasis and then go back to the steady state. However, in the leukemic condition, this myeloid regeneration pathway is constitutively activated by the leukemic stem cell, and then they constantly overproduce this myeloid bias 2 and 3, and then making MPP4 making more myeloid lineage to overproduce the myeloid cell, which lead to myeloid leukemia. So by dampening, I just showed that by dampening this inducible regeneration pathway, we can also kind of dampen the myeloid disease progression. And my second part is when I studied this MPP3 expansion in the myeloid leukemia and the in general steady state thing, actually I found that subset of the MPP3 has a very high endoplasmic reticulum volume. So you can see here the representative electron microscopy, and then this white arrowhead showing that this very dense low ER volume in the cytoplasm. And it's only subset of the MPP3. You can see around like 1/3 of MPP3 has this high ER volume in the cytoplasm. So first I want to check that whether this is true. So I use a KDL, which is an ER marker, and then do the immunostaining. And then yes, some of the MPP3 has a very high ER volume. And I can also confirm that using the ER tracker staining. MPP3 has the most ER volume among other HSPC population. So I want to draw your attention to this morphology a little bit more, because when we first saw this EM picture, we are very striking because this morphology almost looks like the antibody secreting plasma cell. You can see the plasma cell has a very dense area throughout the whole cytoplasm, and the MPP3 was the same. So this makes us to think, oh, maybe MPP3 has a secretory activity. So, I mean, clearly, like previously, we think that HSPC, stem and progenitor population, they just sit there quietly doing not much, only they will be activated, they need to make the more cell. But this was a kind of interesting idea that, oh, MPP3 could actually do some more secretory activity to do more active role in the hematopoiesis. So when I studied this thing, actually, the David Baltimore group at Caltech, they also showed that HSPC population can also secrete cytokine upon inflammatory condition. So I was wondering whether actually MPP3 was a kind of the major population can do that. So I isolate this cell population and then use the same inflammatory condition the David Baltimore group used, LPS and PAM3 CSK4, and then culture them for 24 hours. So basically collect the culture supernatant to measure the secreted cytokine. And this is basically the bar graph showing the intensity of the secreted cytokine with or without the stimulation. And this yellow bar is a mean value. So you can see that without the stimulation, all this HSC, MPP3 and 4, or even GMP, they don't secrete much. But upon stimulation, MPP3 has the most robust secretory activity. And to see a little more detail about what kind of things the MPP3 secreting, so this is 4 unstimulated and stimulated each replicate. And surprisingly, some of the things like the TACI or the galectin-1 is decreased upon stimulation. And upon stimulation, actually, as we expect, a lot of pro-myeloid cytokine or pro-inflammatory cytokine is increased in this inflammatory stimulation, such as IL-6, MIF-1 alpha, MIF-1 beta, GCSF, and then MIP2, all these things. So because we studied this as with the high ER volume in the MPP3, I asked whether actually high ER volume is associated with a higher secretory activity. So I use the ER tracker to distinguish the ER high versus ER low MPP3, and then again culture them, stimulate, culture them, and then measure their cytokine secretion. And clearly, without the stimulation, both ER low and high, they don't secrete much, but upon stimulation, clearly ER high MPP3 have more secretory activity, showing that there is a correlation between high ER volume and secretory activity. So to have a better idea about this secretory MPP3 subset, I did a single-cell RNA-seq on the whole MPP3. And there's a total like 13 clusters, but we could categorize this into the 3 major clusters. One is an immature cluster because it clearly showing more like HSC gene score highly compared to other cluster. And then there's a myeloid primed cluster because they express more like GMP gene score highly. And then there was an intermediate metabolically activated population. From this data, what we've kind of focused on and then more found interesting is this myeloid bias, myeloid primed subset actually expressing FcGR3. So FcGR3 encodes Fc gamma receptor. And in our field, Fc gamma receptor is the one used to marking the downstream granulocyte macrophage progenitor GMP. So actually, we didn't expect that FcGR3 will be expressed in the MPP level because this one is really marker for the from GMP to downstream all the myeloid cell marker. So first, I want to check whether this gene really making the protein in the MPP3. So I stained the Fc gamma receptor, and surprisingly, actually around 30 to 40% of the MPP3 expressed this Fc gamma receptor on surface. And more interesting was this Fc gamma receptor positive MPP3 was mostly EL high MPP3. And then vice versa, if I isolate the EL high MPP3, they are mostly Fc gamma receptor positive MPP3. So basically, this Fc gamma receptor expression is overlapping with the EL high MPP3 population. So again, as I say, this Fc gamma receptor, we always thought that as a marker for the downstream GMP. I asked whether this EL high Fc gamma receptor positive MPP3 is actually transitional population toward downstream GMP. So to test that, I did a bulk RNA-seq on this population. And as we expected, this EL low MPP3, which is Fc gamma receptor negative MPP3, was more close to the immature HSC population. And EL high MPP3 was more close to downstream GMP, showing that in the molecular level, EL high MPP3 is more transitional population toward the GMP population. So to test this molecular data in the more cellular level, I performed the sublethal transplantation. So in this sublethal transplantation, basically the readout is more immature cell, they can contribute more donor chimerism. And then they are more mature cell, they less contribute to the blood production. So HSC serve as a passive control. And clearly this EL low MPP3 has most robust donor chimerism. And then EL high MPP3, they barely contribute to the donor chimerism. And it's normal that usually MPP in the transplantation setting, they cannot maintain their blood chimerism in the long term. So, but clearly this data confirming that the molecular data that EL low is more immature population, and then EL high is more downstream, little more within the MPP3 downstream mature toward the GMP population. Then next question is whether this EL high and EL low MPP3 is interchangeable or they have some hierarchical relationship. So first, I cultured these two populations in the in vitro, and clearly you can see that this Fc gamma receptor negative MPP3 can make a EL high / Fc gamma receptor positive MPP3 in the time-dependent manner. However, this Fc gamma receptor positive MPP3, they don't maintain as an MPP3, and then they just quickly differentiate into the myeloid progenitor, downstream myeloid progenitor population. This was the same in the in vivo situation. So I do the infusion assay, I put these cells, and then clearly negative MPP3, similar to the in vitro system, they can make the Fc gamma receptor positive MPP3 in the time-dependent manner. But I'm not showing here, but Fc gamma receptor positive MPP3, they cannot make MPP, but they just making the more downstream myeloid progenitor, in particular GMP. So there's a lot of data to characterize these two relationship between these two populations, but I will just give you a summary data showing here that, so this from the HSC, EL low MPP3 is made, and then this EL low MPP3 can make another EL low MPP3, and also EL high Fc gamma receptor positive MPP3. However, this EL high MPP3 can only directly make GMP, but cannot make either MPP3 or, Fc gamma receptor negative MPP3 or even positive MPP3. So this data clearly suggests that this EL high Fc gamma receptor positive MPP3 is a myeloid bypass mechanism. So basically they don't go through the CMP, and then they just directly making the GMP when they need to boost up the granulocyte macrophage production. So because we have this EL high MPP3, they are really going to make the GMP fast, not going through the CMP, and then they have high EL volume. I asked whether actually this ER high MPP3 secretion also promote to production of the myeloid cell. So to test that, I used actually MPP4 as reference point because it's a more lymphoid bias. And because MPP4 doesn't have Fc gamma receptor positive proportion, I also use MPP3 as a total. And I collect the supernatant from this cell and then put the HSC into this supernatant to see how much this supernatant support the HSC production of the myeloid cell. And you can focus on this Fc gamma receptor and MEK1 positive myeloid, mature myeloid cell. And clearly, without the stimulation, when MPP3 also don't secrete that much, the HSC culture in the basal media or MPP3 supernatant or MPP4, they don't really make much myeloid cell. However, when HSC was cultured in the supernatant stimulated with the LPS and PAM3, which means MPP3 secrete quite a lot of the pro-inflammatory and pro-myeloid cytokine, HSC will robustly make the myeloid cell in this culture condition, unlike the MPP4, clearly showing that MPP3 secreted cytokine can promote the myeloid cell production. I'm only showing here in the HSC, but when I test in the other population, MPP3, MPP4, all these MPP3 supernatant can support the production of the myeloid cell from this population. Then what about the in vivo condition? It can be also happen in the in vivo. So I know this, so we devised a staining scheme to distinguish MPP2 and 3 and 4 in the bone marrow. Because of the limited number of color we can use in the confocal, I know this resolution is not very good, but still we could distinguish at least MPP2, 3, HSC, and then MPP4 GMPs together. And what we found is MPP3 is found in the kind of vicinity of the other HSC population. And recent paper from the Daniel showing that actually MPP is a migratory population. So we expect that probably this MPP3, when they are secreting, they can kind of move toward the other HSPC, and then maybe can affect on their lineage output by secreting pro-myeloid cell population. So if I kind of summarize this so far, then in the steady state, HSC produce actually smaller portion of the MPP3. But even this smaller portion of MPP3, there's just several molecular subpopulation. Especially there is myeloid prime, distinct secretory activity which have high ER volume and then expressing Fc gamma receptor in their surface. And inflammatory condition, this inflammatory HSC actually overproduce MPP3, and within this MPP3, this ER high and then Fc gamma receptor positive MPP3 secrete the cytokine which will work on MPP3 itself, and then also HSC and MPP4 really to push this cell to the myeloid lineage. So this showing that secretory MPP3 is really the enforcing the myeloid lineage trajectory in this stress condition. Then I want to go back to the myeloid malignancy condition because it's already shown that MPP3, I already showed that MPP3 is highly expanded in this condition. So is this secretory MPP3 has a special role in this driving the myeloid disease again? So when I test this MPP3 with the ER tracker and the Fc gamma receptor, clearly ER high MPP3 is significantly increased in this MPP3. But more interestingly, this Fc gamma receptor positive MPP3 was specifically increased in the MPP3. So basically this MPP3 expansion into this CML mouse model is all driven by this Fc gamma receptor positive secretory activity. So to have a better idea of this Fc gamma receptor positive ER high MPP3 in the BA context, I again did a single cell RNA-seq. And as we show in the previous Fc gamma receptor staining, in this single cell RNA-seq, there is an expansion of this FcGR3, which encoding Fc gamma receptor is hugely expanded. But more interestingly, I found is in this Fc gamma receptor positive MPP3 expanded in the leukemic MPP3, there's a leukemic specific subpopulation. So what's the molecular characteristic of this leukemic specific population? And the CAG pathway analysis showing that basically this leukemic specific population is they are metabolically activated, like protein processing in the ER is increased, and then glycine serine threonine metabolism, glutathione metabolism, and then biosynthesis of amino acids is all up. So because clearly, interestingly, this leukemic specific subpopulation exists in these BA mice, and then these BA mice, especially, it was increasing the EL high, which is a more secretory MPP3 subset. So I asked whether in this leukemic condition, this secretory subset has increased secretory activity. And unlike the inflammatory condition, if you remember, even without stimulation, MPP3, they don't secrete much. Only when there's a stimulation, actually they can secrete more compared to other HSC or MPP4. But interestingly, in this leukemic condition, without any stimulation, actually MPP3 constantly secrete certain set of the cytokine, including IL-6, NIFON alpha, lipocalin, and then some galectin and granzyme B. So it was very interesting because in this, probably this metabolically activated MPP, leukemic specific MPP3 will drive this constitutive secretion of this cytokine. So the next obvious thing was to test whether this secreted cytokine can actually promote more or exacerbate the disease condition. So I picked the galectin-3 as one of the cytokine because galectin-3 is shown to be high serum level in patients with cancer, not only the hematologic malignancy, it's all different kind of cancer. And also it's a poor prognostic factor for the AML patient survival. And most importantly, it was shown to increase Wnt beta-catenin signaling. So to remind you, I showed that in my first part of the talk, the Wnt signaling is very important. It's the driving force to expand the MPP3. So I was asking whether actually this galectin-3 secreted by the leukemic MPP3 will driving more increased Wnt signaling, so can expand more MPP3, which make the disease worse. So to test that, I crossed these BA mice with the galectin-3 knockout mice. And I know there's a lot of graphs, but I will just walk you through for the key data. So in the MPP3 compared to control, MPP3 is hugely expanded, already shown. And then with the galectin-3 knockout, clearly MPP3 expansion was a decrease. And especially this Fc gamma receptor positive MPP3 was significantly specifically increasing this CML mouse model. And then without the galectin-3, it's a decrease. And then also downstream GMP was a decrease. And then also the mature myeloid cell was a decrease. So clearly this data showing that cytokines secreted by the leukemia MPP3 expand the EL high and then Fc gamma receptor positive MPP3. Then what about if I reduce this, then disease progression also decrease. So I crossed these mice with the BA and then monitor their survival. And more interestingly, compared to even BA, not only homo, but het mice also have a survival benefit. That was actually quite surprising, but this is really showing that reducing the cytokine secreted by the leukemia MPP3 can attenuate the disease progression, probably through the lower dampening the expansion of the leukemia MPP3, so downstream GMP and then mature myeloid cell. So basically in the leukemic condition, similar to the stress condition, this leukemia HSC or pre-leukemia HSC overproduce this MPP3. And within this MPP3, there's already metabolically activated subset of high EL volume, Fc gamma receptor positive MPP3, which constitutively secrete a cytokine which will act on the other MPP3, make them induce secretion. And also this all together secreted cytokine from the MPP3 will act on the HSC and MPP4 other population to make them to really produce the myeloid cell. So this Fc gamma receptor positive secretory MPP3 function as a myeloid amplification compartment and clearly driving force for this leukemia. So hopefully I kind of convince you that like MPP3 is really the cellular driver of the emergency myelopoiesis. So everything in together in the steady state, HSC has high notch and low Wnt, so have relatively small myeloid bias MPP2 and 3, and more lymphoid bias MPP4 to have the balanced lineage output. However, in the stress or leukemic condition, so I will just more explain to the leukemic condition, this HSC, basically leukemic HSC hijacking this inducible myeloid regeneration pathway, and then have consecutively low notch and high Wnt activity, which will always overproduce this MPP2 and MPP3. And even within this MPP3, there's a metabolically activated secretory MPP3, which will keep secreting pro-myeloid cytokine, which will act on other MPP3 population and also HSC and MPP2 and HSC to really force this all HSPC compartment to drive to make the myeloid cell. So this secretory active MPP3 in particular is really the passive reinforcement compartment to produce the myeloid lineage trajectory. So this is like kind of my basic starting point for my new lab. And then I will briefly introduce just one slide, what's going on in my current lab. So someone who may be interested to collaborate with me, then we can talk more. But so I show that in using the CML mouse model, which is a BA mouse model, this Fc gamma receptor passive MPP3 just specifically increased. So in this CML mouse model, this Fc gamma receptor passive MPP3, which is basically making the GMP, the one driving force of this MPP3 expansion. It kind of makes sense because the CML mouse model is characterized by the overproduction of the granulocyte. And what I found is, very interesting these days is in the PB model, which is the polycythemia vera, it's another myeloproliferative neoplasm disease model, which is characterized by the overproduction of the erythroid cell. Again, in this MPM model, MPP3 is also significantly increased. But interestingly, in this condition, this expansion is driven by Fc gamma receptor negative MPP3. So because I was more focusing on the GMP and the granulocyte macrophage lineage, I didn't show the data, but basically this Fc gamma receptor positive MPP3 is directly going to GMP, and Fc gamma receptor negative MPP3 is making more erythroid lineage. So this data clearly showing that distinct MPP3 subset can differentially contribute the different MPN according to their expansion of the mature myeloid cells. So CML model, where the granulocyte is overproduced, this positive MPP3 is hugely specifically increased. And this polycythemia model, when there's erythroid overproduced, this erythroid potential Fc gamma receptor negative MPP3 driving this expansion of this lineage. So my question is now, I showed that notch and Wnt increase the Fc gamma receptor positive MPP3 production. Then what increased this Fc gamma receptor negative MPP3 from the HSC? So I'm studying what kind of signaling will drive this erythroid potential Fc gamma receptor negative MPP3. And also I showed that there is a hierarchical relationship between this Fc gamma receptor negative and positive MPP3. So negative MPP3 can make the positive MPP3, but not the vice versa. So I'm studying what's the transition, what signaling regulate this transition from the negative to positive MPP3. So if I know that actually in this CML mouse model, I can make positive MPP3 going to less, negative MPP3 going to less positive MPP3. So reduce the production of this overexpansion of positive MPP3. And in the PB model, I can make this negative MPP3 going to more positive MPP3. So can reduce the expansion of this negative MPP3. So by knowing that I can kind of target two different diseases with the one targeting one signaling pathway. So this is a currently ongoing project in my lab. And then I'm thinking maybe NF-kappa B may be involved in my first question. And then second question is maybe unfolded protein response may be involved in this transition. Okay, with that, thanks for your attention. And then I'd like to thank my previous mentor, Emmanuel Passaget, and then Passaget lab member who make me give all these foundation for my new lab, and my current lab members, and then my funding source. And then again, thanks for your attention. And then I'm happy to taking any more questions.
Great.
Hi,
Thank
Anna.
you so much. Thank you so much for a wonderful talk.
Yeah.
We have a couple of questions populated in the chat. Adil, do you want to ask or modify your question? See if he's still there. So Adil was wondering about the ligands during stress. What, you know, you're showing the markers on those cells, on the MPP3 cells. What about ligand expression that might be driving the shift in hematopoietic fate? I think that's what he's asking here.
Sorry, so when you say ligand means?
Well, so you have soluble molecules that are interacting to determine that fate.
Yeah, so clearly in the, I didn't specifically look at the stress condition, but more in the myeloid malignancy condition. Clearly, I was wondering what caused this lower notch activity in the stem cell. So first thing we could find is actually Notch1 receptor is less expressed in the leukemic stem cell. I mean, still we don't know exactly what caused the downregulation of this Notch1 receptor on the leukemic stem cell, but at least one factor is because Notch1 receptor is reduced on the surface, so they can receive they cannot get the same amount of the notch signaling by the notch ligand provided by the microenvironment. So that's the one factor clearly driving the lower notch in the leukemic stem cell. And then for the Wnt, we found that actually in the leukemic condition bone marrow fluid, so in the bone marrow microenvironment, the Wnt inhibitor DKK1 is increased in the leukemic serum. So clearly, I mean, I bet there's a lot of so many different things regulating those, but one factor is in the leukemic condition, DKK1 was increased, so they can probably have a more reduced Wnt signaling from the, again, microenvironment. So those are clearly a contributing factor. But I bet there's so many other things to lower the Wnt, I mean, increase the Wnt signaling and then lower notch in the leukemic condition, and probably similar to the stress condition.
Great, thank you. One other question, Vulehu, I'm going to mispronounce it, Vulehu. Do you want to ask your question? You can unmute yourself.
Oh, thank you. You have a great talk. So,
hi, Tran.
Hi, Yoon-A. So, my question is from the RNA-seq data of the ER high versus ER low NMP3. Do you see any change in the expression of the hematopoietic transcription factors or any transcription factors known to regulate the expression of signaling molecules involved in Notch and Wnt pathways?
Oh, so you mean the leukemic condition, right?
I remember you have.
Yes, so I have steady state MPP3 single cell data and then leukemic MPP3 single cell data.
Probably that one. Yeah.
Yeah, so I didn't actually specifically looking at those signaling gene expression because it, you know, the single cell is the depth is a little low, so it wasn't actually the top one is a prominent there. But and I think the notch and Wnt signaling is more the change is happening in the HSC level. That's why when there is a high Wnt or low notch in the HSC, they will produce more MPP3. I mean, it doesn't exclude that MPP3 also have a more like high Wnt signaling. I mean, clearly my beta-catenin, nuclear beta-catenin level showing the higher in the MPP3 in the leukemic condition. But one thing I want to mention is we actually look at the bulk RNA-seq data in the human patient sample, and then we could see that there's a relatively low notch signaling and the high Wnt signaling in the human patient sample RNA-seq data. But we didn't have a specific resolution of in the MPP3 level. So we think this notch and Wnt signaling regulation is not only limited to the mouse, it's also in the human system as well. Yeah.
Yeah, thank you. And I'm sorry, I think there's someone.
No,
Anna,
please.
do you want to go ahead?
No, please finish your questions.
Yes, and then when you stimulate the ER high versus ER low MMP3 and with the LPS, PAM3 and combo to measure the cytokines production, and you found the ER high produce more cytokines. So I just wonder if is that because the cells have more receptors on the cell surface to respond to the stimuli, or is that the receptors stay the same, but the ER high MPP3 have the high capacity to transmit the signal, or something happened in the transcription factor?
Yeah, so that's a very good question. And then actually we haven't checked that whether this the toll-like receptor 2 and 4 are differently expressed in the EL high MPP3 versus low MPP3. So it could be both. Yeah, I cannot say because I didn't check, but then it could be EL high MPP3 also express more those toll-like receptor 2 and 4. But another thing we are thinking is because they already have the high EL volume, so when they get the same signaling, probably they have more capacity to making more cytokine faster. So that's why I think they, at least they have more equipment to make those protein. But clearly the expression level of the toll-like receptor could be another factor, but we actually haven't checked that. But that's a very good point.
Yeah, the reason I bring up this is because from my study, when GATA2 is low, the cells, myeloid cells, have more toll-like receptors and the capacity to respond to the toll-like receptor signaling. So I just wonder if the ER high versus ER low MMP3 may have different level of GATA2 or other transcription factors like QP1. Thus may skew the cells.
Actually, I think, yeah, the GATA2 level, I didn't say that here, but actually another factor I'm thinking the transition between ER low versus ER high is, I think, GATA2. Because when I did the bulk RNA in this ER low and ER high, the ingenuine pathway analysis is showing that one of the top regulator maybe regulate these two different populations. One of them was a GATA2. So I also think that GATA2 level probably different in these two populations. And then I suspect that because of the GATA switch or those Emery Bresnik discover, probably GATA2 high will be more stained as a ER low MPP3, and GATA2 level low will go to more ER high MPP3, I suspect. Yeah. I think GATA2 level will regulate this different subset. Yes.
Yeah, thank you.
Great. Anna, did you want to ask a question or?
Yeah. Great talk, Yoon-A. Great to see you.
Thank you. Hi. Happy New Year. Good to see you.
So I just had a question, and I struggle with these definitions sometimes too, so forgive me for this. But so where, like, so the MPP3s, they're like close to a GMP, right? Like, so are they do they have lower SCA expression levels? And then so that brings me to my next question, which is, are there secretory GMPs? Like, is there, where do you draw that boundary?
Yeah, that's actually a very good question, because like clearly, I mean, so the SCA1 expression, but clearly, the overall now secretory MPP3 only, but like GMP and MPP3, the only difference is the SCA1, right?
Right.
So clearly, because we use the SCA1 as the marker, immunophenotypic marker, still EL high MPP3, because it's a part of the MPP3, SCA1 level is way higher. It's not like the DIN, it's like same high as the SCA1, but in the molecular level, as I showed in the bulk RNA-seq, they are actually very similar to the GMP. More right. EL high MPP3 is more GMP. So as you can say, yeah, if you say, oh, how do you know this is not part of the GMP, but like MPP, because only SCA1 expression, yeah, it's all continuum.
Right.
So yeah, I mean, only based on this kind of SCA1 expression, we kind of, because we said that LSK population is LSK, so we call it MPP3. But when I check the cell cycle status or the proliferation, EL high MPP3 is more similar to GMP, not the EL low MPP3. So they are very similar to GMP in the many ways. So it's a,
right.
Yeah, you're right. Like it's.
Do you also get GMPs that secrete like this? I mean, that could be, I mean, do you have secretory GMPs? And then my second kind of follow-up question to that is, because you did, part of your secretory or these ER high GMPs is that they, this idea that they have a bypass to the GMP, right?
Yeah.
And those, sorry, I had to step out for a second, but you did these transplant assays where you transplant high numbers and you look downstream, right?
Yeah.
How many days you did? Was it 3 days or 2? How many days after did you look in the bone marrow for those?
So I look at both 2, 3, and 4. You mean the infusion assay?
Yeah, where you put in like a high number of the ER high, and then you look to see what progenitors are making the bone marrow. I mean, is it possible that it's just too fat? Like they're too close. to see that it's going through? And do they not make MEPs, or can they, like, in your JAK2 context, do they
Yeah.
then make
Mhm.
MEPs? Like, how does that work?
Yeah, that's a very good point. But at least when I look at this ER high MPP3, they only make GMP. They didn't make the MEP. So that's
Okay.
why we say, oh, this in the in vivo situation, this ER high is really only making the GMP. But the ER low, they can make both CMP, GMP, MEP. But
Okay.
that's a good point. I haven't looked at it, but in the JAK2 model, maybe this ER high MPP3, they also kind of toward the more aristotle lineage, they can making more MEP. But I totally agree. It's a very, I cannot just say, oh, ER high MPP3 is MPP3. It could be someone can call this as a more like immature GMP, right?
I mean,
So
it doesn't
yeah,
matter.
it's hard to draw the line. Okay, this is because the only difference is kind of SCA1, but in the molecular way, cellular way, ER high MPP3 is very similar to the GMP. So unless we figure out what's the function of SCA1 in this ER high MPP3, different from the SCA1 in the GMP. I mean, GMP not really having the SCA1, but until we have a clear answer for that, it's hard to say, oh, they are totally different population, right?
Right. I mean,
Yes.
maybe the secretary, that was my question about whether GMPs have